Hello and welcome to another video. In our last video, we discussed the process of mitosis and its importance in organisms. Today, we're going to stick to the topic of cell division. However, we will be discussing the process of meiosis and its effects on humans. If you missed my previous video on mitosis, feel free to go back and watch it. That way you have a deeper understanding of the terms that may be used throughout this video. To start, we must ask the most important question. What is meiosis? Meiosis is a process in which one diploid parent cell divides twice, resulting in 4 daughter haploid cells which only contain half the chromosomes of the parent cell. Meiosis occurs within the sex cells of an organism, for example, eggs in women and sperm in men. It is also a necessity for the reproduction of human life. Before we go into any further detail, here are some of the key terms you may want to know before we start. A gamete is the name given to a reproductive cell. In males, it is called sperm, and in females, it's called ova. Zygote is known as the fertilized egg cell produced when a female gamete and a male gamete combine. Random fertilization is something that takes place throughout the entire meiosis phase, and it is basically the difference between every sperm cell and every egg cell and how they will have different genetic variations. Random fertilization is known as genetic variation 1. Crossing over is the process of chromosomes exchanging little pieces of DNA, and this happens in prophase 1. Crossing over is also known as genetic variation 2. Independent assortment takes place during metaphase 1 and is the process in which the homologous chromosomes line up on the metaphase plate in complete random order. This means that when the cell divides in two, the chromosomes will be split completely randomly. This process is known as genetic variation 3. Meiosis follows a very similar process to mitosis. In fact, it still follows the IPMC acronym. However, meiosis actually follows this entire process twice, except for interphase, as the cell needs to divide twice but only needs to duplicate the genetic material once. Once again, I'll be using a homemade model to assist in my explanation, and I must remind you that it is not to scale and is only a simplified representation of the meiosis process. My model will consist of 1 to 4 chromosomes, as there are so many in the human body and I don't want to risk confusion. Now that's all cleared up, let's jump right in. Interphase is the first step of meiosis and is used very much like a preparation stage. The main events which occur throughout this process are the synthesis of a variety of necessary components, including nutrients, regulators, and enzymes required for the upcoming replication process. DNA replication, also known as the chromosomes doubling, and the buildup of nutrients which will be used later in the process for the development of meiotic spindle fibers. In the beginning of this stage, there are 23 pairs of chromosomes, but due to the replication phase, it will conclude with 46 pairs and 92 chromatids. Throughout prophase 1, X-shaped structures are formed when chromosomes are cloned. Each chromosome is made up of 2 sister chromatids with the same genetic material. After then, the chromosomal pairs may exchange little bits of DNA in a process known as crossing over. This process will affect the rest of the phases through meiosis, as the genetic material is no longer identical to the original diploid cell, which is why the chromosomes are given the name homologous chromosome. Throughout the conclusion of prophase 1, the membrane covering the cell's nucleus dissolves, releasing the chromosomes. During this entire phase, there are still 46 pairs of chromosomes. During metaphase 1, on the cell's equatorial plane, the chromosomal pairs are arranged side by side. Shortly after this, the meiotic spindles begin extending from the centrosomes, which are now located at the cell's opposing poles. The meiotic spindle fibers will lengthen and shorten in search to attach one chromosome from each pair. This process will consist of 46 pairs of chromosomes, and this phase will also include the process of independent assortment. Anaphase 1, much like mitosis, is the phase which involves the separation of genetic material. The meiotic spindle will pull one chromosome to one pole and the other to the opposite pole of the cell. The sister chromatids remain together throughout meiosis 1, in contrast to mitosis and meiosis 2. The homologous chromosomes on each side of the pole will still contain the little bits of DNA which were previously exchanged during prophase 1. Throughout this stage, there are still 46 chromosomes, however, they have been separated to the opposite ends of the cell evenly in separation and preparation for the division of the cell. This phase will also include random segregation. Telophase involves the homologous chromosomes reaching the opposing poles of the cell in whole. A complete set of 23 chromosomes will congregate at each pole of the cell. They are now then slowly surrounded by a membrane that divides into 2 new nuclei. This is the last stage before the final separation into 2 daughter cells, and there are still 46 chromosomes in total. The original cell's cytoplasm is now split into 2 daughter cells, as the cell goes through a process known as cytokinesis. These daughter cells will now start the second phase of meiosis. We now find ourselves in the second step of meiosis. This begins with prophase 2. During this phase, there are currently 2 daughter cells, each of which has 23 chromosomes. The chromosomes are condensed into distinct X-shaped structures in each of the 2 daughter cells, where they are then released due to the nucleus membranes breaking down. During this process, the centrosomes are duplicated and prepared to navigate their way to the opposite poles of the cell. We'll now move on to metaphase 2. In each of the 2 daughter cells, the chromosomes will align end to end on the equatorial plane of the cell. Whilst this is taking place, the centrosomes should have arrived at the opposite poles, where the meiotic spindle will begin to lengthen and shorten again in search for the chromosomes. Once the meiotic spindle has found the chromosomes, it will attach to it at the kinetochore. During anaphase 2, the meiotic spindle will begin to shorten, splitting the centromere and resulting in the sister chromatids being divided and pulled to separate poles of the cell. The chromatids will stay on either end of the cell in preparation for the formation of the nucleus and the division of the cell. This phase will also include random segregation. We are now nearing the end of this process. This stage is telophase 2 and consists of the sister chromatids arriving at the separate poles. Soon after this, the nuclei will begin to form around the 2 sets of chromatids. Cytokinesis 2 is the final stage of meiosis and involves the division of the two daughter cells, creating 4 granddaughter haploid cells. These cells will have half of the original genetic material that the first diploid cell had. For example, the diploid started with 46 chromosomes and 92 chromatids, and the granddaughter cells will have 23 chromosomes each. This is the conclusion of my video. So once again, thanks for watching, and if you like this video, please like and subscribe and hit that bell button so you are notified when I post.